Okay, our next speaker is David Rocco from UNC Chapel Hill. Once I find his presentation. It is here. I know it is. David, where are you? Here we go. You good, David? Is that okay?
All right, I'd like to thank everyone for coming to listen to my talk, and I'm really excited to be here, and I'm really thankful for the opportunity to speak here as well. So I'll be telling you about a potential role for integrin activation and aggregation in immune thrombocytopenia, done in the lab of Dr. Wolfgang Bergmeier. So just a brief overview of the relevant platelet functions that are important to this talk. We have initial platelet tethering to the subendothelium, which is mediated by GP1B9. We also have signal transduction, which activates the small GTPase Rap1, and with in coordination with the cytoskeletal adapter protein TALEN1, this mediates activation of the integrin alpha-2b beta-3, and this facilitates firm platelet adhesion and aggregation. Interestingly, this first adhesion receptor GP1B is also a target for autoimmunity or auto antibodies that recognize the alpha domain, and this can lead to translocation of neuraminidase, desylation, and this can lead to platelet clearance. So this is what I'm going to talk about. We can actually use a polyclonal mixture of antibodies against GP1B alpha and inject this into mice, and you can see in this video that this forms these large aggregates of platelets in the liver sinusoids, and they kind of form they form these large aggregates that then get rapidly phagocytosed by the macrophages, the Kupffer cells, which are in the liver, and we can also use this antibody in vitro. And you can see that in comparison to some stills from this video, you can see that before addition of this antibody, you kind of have circulating and resting platelets visualized by this discoid morphology. And after adding this antibody, you can see that you kind of get similar aggregates of platelets in which they almost look like quiescent platelets, as they still retain this discoid morphology. So to investigate this in vitro, we isolate the platelet-rich plasma, or PRP, from mice. We can then add in our anti-GP1B alpha antibody, spin these platelets down into a platelet pellet, and upon resuspension, we get these platelet aggregates that over time eventually settle to the bottom of the tube. Before this happens, we can actually take these aggregates and image them via confocal microscopy, as seen in this image. And then we can also, after they settle, we can take the supernatant, and there are some residual platelets remaining in suspension, and we can quantify this by flow cytometry. On the right here, you can see that with just addition of PBS, you kind of get this nice, very cohesive platelet population on the flow cytometer. When you add in this anti-GP1B antibody, you see the presence of these smaller aggregates that are forming, indicative of this streaking to the top right, which shows an increase in size. And when you knock out this receptor, again, you see something that looks very similar to the control, in which none of these aggregates are forming. So being that these platelets are sticking together, we wanted to look at the main process that facilitates platelet aggregation, as I mentioned, mediated through TALEN and alpha-2b beta-3. So if we inject if in the presence in vitro of an alpha-2b beta-3 antibody, which blocks this integrin function, in the presence of the GP1B alpha antibody as well, you can see that blocking this integrin function really doesn't have an effect on the amount of platelets that are clustering together and falling out of suspension. Additionally, when we use mice that are lacking TALEN1, you see something similar, albeit there might be a small difference at an intermediate dose, but overall it seems like there's really not much contribution of integrin activation and aggregation to this process, so going forward I'm going to call it cluster formation. So going upstream of this, as I mentioned, RAP1 in coordination with TALEN is what activates integrins. And so we wanted to see if RAP1 at all is involved. And I'm trying to establish a connection between GP1B and RAP1, which has not been well studied. So we can actually fish out the active form of RAP1 from the total pool of RAP1, and you can see that the presence of centrifugation alone and with resting platelets, you don't get very much RAP1 activation. But after adding this anti-GP1B antibody and spinning them down, you can see that you get some modest RAP1 activation here. Using mice that are lacking RAP1, you can see at these intermediate doses, there seems to be a modest yet significant effect in reduction of platelet cohesion. Although this kind of this difference gets attenuated when you raise the dose high enough. Suggesting that RAP1 might be having a role in this, although albeit a subtle role. So just to conclude, I've shown that anti-GP1B antibodies cause these rapid platelet clusters that form and get sequestered to the liver, and this antibody can cause this both in vitro and in vivo. This process of platelet cluster formation occurs independently of integrin activation, but may involve the small GTPase RAP1. So I'd like to further investigate how are these platelets actually sticking together if it's not through these beta-3 integrin. And also, is the reduction in platelet clustering just due to an increase in platelet size, which is seen in these RAP1 null platelets, or if this is actually a signaling defect that is occurring. And lastly, I'd like to know how these platelet clusters are phagocytosed by Cooper cells. There is some literature suggesting that changes in surface sugar structures on platelets is involved in this, but I'd also like to know if RAP1 is contributing to this process as well. So I'd just like to thank the members of the Bergmeyer Lab. I don't have an acknowledgement slide. And I would also like to thank all of you for coming again, and BioRender for the images that I made.
Yeah, nice talk. Did you rule out the possibility that the antibody was acting through the FC receptor? I guess the mice don't have it, so it was...
Yeah, so the question is this mechanism occurring through the FC mediated process? And actually, so you can use a Fab2 fragment of this GP1B antibody, which doesn't have the FC portion of it, and you do not see a reduction of this process.
You have the same thing. And do you know if the treatment with the antibody increases surface phosphatidylserine exposure in the platelets?
Yeah, so in my hands, I haven't seen an increase in PS exposure. We know that PS exposure requires a very high calcium mobilization that also needs to be sustained for a longer period of time. Being that this process happens very quickly, and we also don't see surface markers such as integrin activation with John A binding or also P-selectin exposure, which is kind of indicative that these platelets are not really proceeding through their full mode of activation, which would then lead to PS exposure.
But there might be other ways to activate the scramblase without activating the entire platelet.
Yeah, that is something really interesting that we could look into. We have someone that is also investigating phosphatidylserine exposure on platelets.
Great talk. Also nice to see a fellow Cooper cell mention. There are reports that polyclonal GP1B antibodies cause John A binding as well as full platelet activation. So to clarify, you use polyclonal?
Yes.
So how do you reconcile then the discrepancy of those reports?
I mean, it could be like a methodology thing, but you know, in our hands, we've looked in vivo as well as with the flow cytometer, and I mean, we do see an increase. It's very, very small, and it's not very indicative of like a full-blown platelet activation. So I would think that like there are subtle modes of activation in platelets. We would have to kind of do some more investigative studies to see that full extent. But there are kind of like intermediate phases of platelet activation in which like, you know, you have different conformations of like the integrins on platelets. So it would be something interesting to look at if we kind of fall in between that range of platelet activation.
Okay, maybe we can chat after.
Sure.